Hey, good afternoon. My name is Jian, Duan. I'm with the U.S. Department of Agriculture Research Service and Beneficial Insect Research Unit at Newark, Delaware. And so today I'm going to talk about natural enemies introduced for biological control of the Amedash borer. But first, I would like to acknowledge all the people that have contributed to the work that I will be presenting today. Many people from my own lab at New York, Delaware helped the project, and also my collaborator Leah Bauer at the USDA Forest Service, Northern Research Station, and many students and technicians from her lab also helped work. My collaborators, Julia Gold and Benjamin Slager from USDA APHIS, Chris Elbow and Roy Van Dredge from University of Massachusetts have helped with the project I will be presenting today. So I assume most of you can recognize this beautiful metallic green beetle, the Amedash borer, often called green menace in the media. It is a very beautiful beetle, but unfortunately it is also very invasive pest that have killed millions, hundreds of millions of our ash trees. And this beetle was probably introduced from China sometime in the 1990s. It was first detected in Michigan in 2002. Now it has been detected in 32 US states. It has caused widespread ash mortality, causing not only economic losses but also cascading ecological impacts. One of the challenges in the detection and control of this pest because it has a very cryptic life history. This video shows a female beetle lays her eggs in the loose bark or under bark crevices. So basically the eggs are not visible at all if you don't look for it. And the eggs normally takes about 7 to 10 days to hatch, and the larvae basically burrow into the flume, feeding and developing under the bark for 1 or 2 seasons. It means it has 1 generation per year, or sometimes may take 2 years to complete a generation. The adult beetles normally don't do a lot of damage. They just nibble on ash foliages. It's the larvae that feed in the cambium tissue. As shown in this slide, the larvae can effectively girdle the tree and interrupting the water nutrient transport of the tree. When a tree infested with even moderate density of the EEB larvae, it can be killed in 2 or 3 years. So over 95% of the life cycles are under the bark are cryptic. Those include eggs and larval stages. Unfortunately, those are the stages that are also susceptible to complexes of natural enemies, including some egg parasitoid and several species of larval parasitoids. As I just mentioned, the adult beetle normally don't do a lot of damage, so are not really targeted by biological control. So why we think biological control of Amedash borer in North America will work? First, some ecological studies in Northeast Asia, the native range of Amedash borer, indicate that there is a complex of co-evolved natural enemies there that keep the Amedash borer population very low. So if you go to Northeast Asia, you occasionally see some ash trees, North American ash trees there being infested or killed, but nothing like the scale of infestation we see in North America. So we believe that the natural enemies co-evolved with Amedash borer in its native range can in fact help us to control this pest. Secondly, our study in Michigan, Pennsylvania, and other states early on showed that although we have a complex of North American natural enemies, such as woodpeckers, and also some North American parasitoids that feed on Amedash borer larvae, but our native natural enemies in general are generalist, and they do not really keep the Amedash borer at the low population levels, at the low densities. So the classical biology control program I'm going to talk about is really to reconnect the co-evolved natural enemies from the Amedash borer's native range and with the Amedash borer in North America, and hope that these introduced or co-evolved natural enemies would result in permanent management or control of this invasive pest. So the EB biocontrol program was started shortly by USDA after the Amedash borer was detected, found in southern Michigan. The first phase of the program involved the farm exploration for natural enemies in the native range of the Amedash borer. So between 2003 and 2007, 3 major parasitic wasps were discovered in China, as shown in this map in northeast China. These include an egg parasitoid and 2 larval parasitoids. Unfortunately, there is no common name for this insect, so you have to bear with me when I use their scientific names, and sometimes I'll give them an acronym. Okay. The egg parasitoid is Obius agrila. We sometimes call Obius. And the 2 larval parasitoids, one is a black-horned wasp, the large one. It's Species agrila. And the smaller larval parasitoid is this one. It's Europid wasp. It's called Tetrasticus planipanisa. Okay. Both larval parasitoid attack late stages of the Amedash borer larvae. I will come back to talk about their biology later. Okay. So these parasitoids were identified and imported to the U.S. and tested for their safety, that is their host range, and then approved by USDA aphids for release in the U.S. in 2007. However, the major releases of these 3 introduced Chinese parasitoids did not really start until 2010, before the aphids mass rearing facility was established. From 2008 to 2010, the USDA ARS expanded the farm exploration to a little bit further northeast to the Russian Far East region, where we also discovered 3 Amedash borer parasitoids. The larger black-red wasp, Antanaculus nigriventris, and also a black-horned wasp, Species galena, and also a new species of egg parasitoids. As of now, only Species galena have been approved for release in the U.S. And this parasitoid was evaluated, is found to be very specific, attacking Amedash borer. So it was approved by USDA aphids in 2015, and the releases started in the summer of 2016. So this slide basically shows you where EEB is distributed in Northeast Asia and where these 4 approved biocontrol agents that have been introduced to the U.S. The 3 Chinese parasitoids were discovered in Northeast China, and one of the approved Russian parasitoid was collected from the Russian Far East near the Vladivostok. So these 3 Chinese parasitoids, as I mentioned, was approved in 2007, and as of last year, at least 1 or more of these parasitoids have been released in 24 U.S. states, as shown in this slide. The Russian parasitoid was only approved in 2015, so really this map did not include the releases of the Russian parasitoid. As of now, the Russian parasitoid was only released in several northeast states, New York, Massachusetts, Connecticut, and Michigan. So not all the release sites have been sampled for their establishment. As of 2015, among the sites that sampled, we found that Tetrastix planipanisa, TETs, as shown in this table, and the egg parasitoid Obius agrelli have established in most of the release sites. However, the introduced Chinese braconid wasp species agrelli has only established in a few locations. So in the following part of the talk, I'm going to primarily focus on the larval parasitoid Tetrastix planipanisa and egg parasitoid Obius agrelli. So I'm going to go over briefly the biology of this egg parasitoid and the larval parasitoid, and whether this parasitoid have any impact in terms of managing the Amadeus borer in North America. Okay, the egg parasitoid Obius agrelli. It is a solitary egg parasitoid. That means each parasitized EB eggs, they only produce 1 wasp. What is really interesting is this wasp reproduce parthenogenically. That means the mother reproduces daughter without males. So far we have not seen any males being produced in the lab. Okay, so all the colony we have are females. So each female can live 4 to 6 weeks and attack 50 to 60 freshly laid EB eggs. It normally completes its generation in about 3 weeks. This video shows a female Obius agrelli is examining the freshly laid Amadeus borer eggs, and once the female decides the eggs are suitable for her baby to develop, she will insert her ovipositor inside the Amadeus borer eggs and deposit the single egg, where the egg will hatch and fit inside the Amadeus borer eggs and eventually develop a wasp. In northeast China, this wasp may have 2 or 3 generations. However, we recently found that this parasitoid, in fact, have a very interesting diapause behavior, which kind of limited how many generations it can produce. As you can see here, a female Obius agrelli is inserting eggs inside the Amadeus borer eggs. Okay. It normally takes several minutes for a female to penetrate the eggshell of EB eggs and deliver her own eggs inside the EB egg. So this is This slide shows you briefly about the life cycle of this wasp. Normally, the wasp emerge in the spring and summer when EB adults coming out of the tree began to lay eggs, and the adult wasp live for 4 to 6 weeks, basically parasitize any freshly laid EB eggs. However, the EB eggs will not be available in late summer, especially in August. So the wasp have to be able to really synchronize well with the EB adult emergence and the EB ovulation period. To overcome the food shortage in late summer and the fall, because there is no EB eggs in the fall, this wasp will begin to produce a diapause progeny. Diapause means it's kind of dormant. It produces to produce progeny that will develop to mature larva stage, but the larva basically will not develop until the next spring. Okay, so the key to establish this wasp is to release the wasp at the right time when EB adults are emerging and ovipositing on ice trees. If you release too early or too late, and this wasp will not have anything to feed on. It only lay eggs inside the hermaphroditic eggs. Okay. And the black eggs here are actually EB eggs parasitized by this tiny wasp, and it contains immature larval parasitoid. And this parasitoid, after overwintering, will emerge to adults. So how to monitor this egg parasitoid? And the parasitoid is very small. It's about 1 millimeter. Imagine to find hermaphroditic eggs under loose bark or bark crevices. So to monitor this parasitoid is also very challenging in the field. We have found that 2 methods are relatively effective in monitoring the parasitism in the field. One is through visual search. So basically, you take a small knife, look for EB-infested trees, and remove all the loose bark. You can reveal the EB eggs. If the eggs are, and then collect those eggs, bring them to the lab. If any of the eggs parasitized by Oobius agrilae, and it will eventually become dark. The other method is through bark sampling. So in the spring or fall, we basically use a draw knife to strip the loose bark off the tree and use a plastic sheet laid on the ground to collect all the loose barks and bring them to the lab, sifting through sieve and dislodging all the eggs. So the eggs will come through the sieve, and then you examine those sifted under a microscope. That's how we monitor the egg parasitism. A long-term study conducted in Michigan by Abel et al. In 2014 indicating that this egg parasitoid, in fact, has established a self-sustaining populations in all 6 Michigan study set. Okay, the blue bar representing the visual survey method. The green bar represents the bark sifting method. Okay, as you can see, there are some differences between really one time. There's some difference in 2012 between these 2 methods. But overall, the bars on the left indicating the parasitism by EEB, the top bar, all the bar at the left, these bars indicating how many trees with parasitized eggs. But overall, the parasitic rate by this egg parasitoid is low. It's between 5 to 20, less than 25%. And also, we did not recover any parasitism in the control study set until 2012. The distance between control and release plot in this study set is about 1 to 5 kilometers. Okay, so indicating that this wasp did not really spread very fast. It takes several years really for the wasp to spread a few kilometers away from where it was released. So now let's talk about the introduced larval parasitoid, the Europhic wasp, Tetrasticus planipanacea. This is a larval parasitoid attacking primarily second and fourth instar, means large, medium to large Amedesh-borer larvae. Okay, each parasitized Amedesh-borer larva can produce between 30 to 170 parasitoid wasps. Okay, so this wasp, in a way, it has a very high reproductive potential, and also a complete generation in about 4 weeks. What is more interesting with this wasp is its development is thermoregulated. It does not have diapause. So as long as the temperature above 13 Celsius degrees, and it will develop, and it can attack Amedesh-borer larvae in the spring, in the summer, or even in late fall. Okay, it overwinter inside Amedesh-borer gallery under the bark. And this slide shows, you see all these like a braid, maggot. These are Amedesh-borer larvae containing, this is the Amedesh-borer larva containing over several dozens of this Tetrasticus planipanacea larvae. Okay, and these larvae overwinter under the bark. In the spring, when temperature reaching 13 Celsius degree above, and they develop to pupa, and then they emerge, they chew a hole through the bark, they emerge to small to wasps, and these wasps began to look for Amedesh-borer larvae under the bark to attack. So really, in a location where the Amedesh-borer have overlapping generations, the larvae available through spring to the summer to the fall, and this was very, very effective. So how to monitor this larval parasitoid? There are people have been using yellow pines traps and cutting down trees and rear the wasp out. What we found actually is quite effective method is to go to the field, find EEB infested trees in the early spring or late fall. At these times, all the larval parasitoid are basically under the bark. Okay, so you remove the bark, you can reveal the EEB gallery and the parasitized and the immature stages of parasitoid larvae. So a long-term study was conducted in central Michigan, where we have 6 sites. Each of the sites was divided into 2 plots. One was released with the parasitoid, and the other plot was not released with the parasitoid. The distance between the control and release plot within each site is approximately 1 to 6 kilometers. Okay, the parasitoid Tetrasticus planipanacea was released from 2007 and through 2010. Okay, and as you can see, this parasitoid was almost detected immediately in the control plot That means it spread very quickly, unlike the egg parasitoids. Okay, but the parasitism rate did not really increase until 4 or 5 years later after the final release. Okay, really until 2012. There's no difference in parasitism rate between control and release treatment. So by 2014, the parasitism rate is around 30 to 35% in both control and release site. What is more important is that what we found in 2013 to 2015, we also sampled smaller trees. We call saplings. So these saplings with the diameter at breast head, the DBH, it's about 2.5 body inch to 2 inches. Okay, really small saplings. What we found that this Tetrastix planipanensis is really, really effective in attacking the medium to large emerald Dutch borer larvae. It caused 36 to 85% of the parasitism. It means killed 36 to 85% of the large to medium emerald Dutch borer larvae. Okay. Well, one of the issue with this parasitoid, however, as you can see from this figure, the tree we sampled here is the DBH is about 7 to 21 centimeters. So I would call small to medium-sized trees. Okay, and the saplings is 2.5 to 5.8 centimeter DBH. So these are small trees. So one of the issues with this Tetrastix planipanensis is that it has a very short ovipositor. The ovipositor is about 1.5 to 2.5 millimeter long. It only effective attacking the emerald Dutch borer in saplings and on small to medium-sized ash trees. As ash grow larger, as shown in this picture, and the Tetrastix planipanensis would not be effective in protecting the larger trees. Okay? That's one of the reasons we think the larger parasitoid, species Galena, collected from the Russian Far East, would be complementary, at least they help to protect large ash trees. So this was species Galena has much longer ovipositor, 2 or 3 times longer than the Tetrastix planipanensis ovipositor. It can reach EB larvae in large ash trees with DBH probably around 25 to 30 centimeters. So this was tested in the quarantine lab for several years for their safety or host specificity, and it turned out it is in fact a very host-specific parasitoid. It only attacked the emerald Dutch borer. So it was approved by USDA for release in 2015, and as I mentioned earlier, it has been released only in several northeastern states, and the evaluation has really just begun. Okay. So now I'm going to talk our native natural enemies. As I mentioned early on, shortly after the emerald Dutch borer was detected in Michigan, Dr. Leah Bauer from Forest Service and other researchers look at our North American native parasitoids. So basically they found the woodpeckers are very, very important natural enemies, feeding on larger emerald Dutch borer larvae, and also there are some complex native parasitoids, as shown here. Also, there are some levels of trait resistance, disease, and competition. Okay? However, none of these natural enemies are sufficiently effective in keeping the emerald Dutch borer larvae and low densities. However, they do a lot of feeding of the Amethyst-borer larvae when the EB population are very high. So the woodpecker predation rate, in fact, is very, very high. If you look at look at the trees that heavily infested with Amethyst-borer, in our study site in Michigan, it's ranged from around 60% to 20%. Okay, but one of the native parasitoid, actually the complex of this group, Entomologist, this group consists of 5-6 species of species. Okay, it is a solitary egg parasitoid. It's beautiful, with a beautiful red abdomen. In fact, it attacked many. It caused heavy Amethyst-borer mortality in the earlier EEB invasion wave. So this Entomologist parasitoid is a solitary egg parasitoid. It lays a single egg on outside of EEB larvae, and the egg hatch and basically attach itself to the Amethyst-borer larvae, basically suck out the juice out of this larvae and eventually cocooned. It may has 1 or 2 generations. However, we recently found that this native parasitoid has a diapause behavior. It means they come out of the parasitized EEB larvae in May or June, but a lot of their progeny will only reproduce 1 generation. So once they reach a cocoon stage, they basically are not emerge to adults until a winter, until the cocoon experienced winter exposure. So this kind of limited its rearing or production because it diapaused. It's only reproduce 1 year, 1 generation a year. So this figure shows you the parasitism of this native Entomologist, okay, in our Michigan study site. So in 2008, 2009, we did not find really almost nothing, no parasitism by this wasp. But all of a sudden, in 2010, it comes. Somehow this wasp has a strong numerical response to the Amethyst-borer density. In 2010 is where the Amethyst-borer population exploded in our study site. So all this group of parasitoid came from nowhere to our study site and it caused 40 to 60% of the EEB larval mortality. Okay, and it's lingering around in our study site even in 2015. Okay. So this native parasitoid, in fact, may have played a very important role in crushing the Amethyst-borer populations in our study site. I'm going to show you the EEB population dynamics in a few minutes later. However, what this parasitoid, again, their native host is some other wood-borrowing insects. When EEB density crashed, they basically move away, move to other hosts. So in the next part of my presentation, I'm going to focus on the impact of these natural enemies on EEB population growth. So yes, we have got at least 2 natural enemies introduced from EEB's native range, established in some U.S. locations. But the next question is, can these introduced natural enemies help us successfully managing the Amethyst-borer populations and protecting our North American ice trees? This figure shows you the Amethyst-borer population dynamics, density changes in 5 years from 2008 to 2015 in in our Michigan study site, in central Michigan. Okay, so really at the peak, the Amedesh borer larva density reached about 40 to 50 per square meter of flume area. Okay, this is a resource-adjusted density. That means this density was adjusted based on the live flume areas. Okay, you saw the big crash of the population from 2009 to 2010, and trying to get back in 2011 and crashed again in 2012. Okay, and ever since, even as of now, the EB density in Michigan has been very, very low. Okay, so you can argue that the crashing of EB population may due to the larger scale of ash mortality in Michigan. However, again, this density is resource adjusted. So yes, a lot of large ash trees being killed by Amedesh borer and reduce the total population. However, the number of the live trees also reduced. That means the live flume area also reduced. So the density, if there's no other mortality factor acting on EEB populations, the density really should not crash. Why Amedesh borer density population crashed in Michigan? So it indicating that other mortality factors, as I just talked about, woodpeckers, the introduced biocontrol agent, and the native parasitoid have played a very, very important role in crashing Amedesh borer population, at least the invasive populations during the peak. So how do we demonstrate? Because as you can see here, there's no significant difference between control, parasitoid control, and release plot. So how can we say that the natural enemies in fact play a role in crushing our Amedesh borer populations and maintaining Amedesh borer population in a low density, so our ash saplings or trees can recover in this study site? So one of the methodology we use is called life table analysis. Okay, for those of you who took insect ecology, so you know how to construct life tables, because the way we sampled the Amedesh borer larvae and associated parasitoid allows us to construct lab tables for the Amedesh borer population in our study site. Okay, so I will lead you through this figure. So the y-axis here is the net population growth rate of the Amedesh borer populations sampled on the ash trees, small to medium ash trees with the DBH 7 to 21 centimeter in diameter. Okay. So basically, based on all the larvae we observed and mortality we observed, and we construct the lab table, we calculate the net population changes, net population growth rate. I mean population changes year after year from 2008 to 2015. Okay. So the blue line represents the population growth rate. Okay, with all the mortalities we observed, including woodpeckers, parasitism by the native parasitoids, as well as the biocontrol agent we released in our study site. Okay, and then after we estimated the net population growth rate with based on our observed data, we removed our introduced agent, Tetrascus plantapanacea, from the lab tables and recalculate the net population growth rate. As you can see, the green line /dashed line. Okay, so when we remove the introduced control agent from the data, we're going to see that there's really not much change in terms of Amedesh borer population growth rate from 2008 to 2011. Only from 2012 we began to see, if we remove the introduced parasitoid, the EB population growth rate is will change, increase in this almost 50%. Okay, the green line. The red line is all the parasitoid removed, the difference between green and red line represents how much our native parasitoid contributed to reduce to the EEB, to the reduction of the EEB population growth rate. Okay, so as you can see, the introduced bowel control agent really did not start playing any role in terms of reducing EEB population until 2012, the green line. Okay, our native parasitoid, as you recall that from previous figure, really play a very important role in 2010 in terms of crushing the peak EEB population. Okay, and still, actually, the native parasitoid play a quite big important role, but the impact of native parasitoid kind of reduced in 2014 and 15. Okay, particularly in 14, the difference between red and green line becomes smaller. So really, the introduced bowel control agent, particularly the larval parasitoids, have begun to suppress the EEB densities, at least to maintain EEB densities in the at low levels. Okay. What is more important, here's the same approach we used to analyze the effect of the introduced bowel control agent on EEB population on saplings. On saplings, the impact of Tetrasticus is much more significant or dramatic than on the larger trees. As again, the blue line is the net population growth rate of EEB with all the mortality factors included. Here, I want to point out that on small saplings, the woodpecker predation is very, very low. Okay, less than 10% EEB preyed upon by woodpeckers. In fact, Tetrasticus is the dominant parasitoid or mortality factors. So the red line is when all the parasitism removed. The difference between red and green lines represents how much the native parasitoid contributed to parasitism of the EEB larvae. Really, in 2014 and 15, all the contributions to the reduction of the EEB population growth rate on sapling trees came from Tetrasticus planipanacea. Okay. So this is the last figure slide I'm going to show you. Basically, again, this is the 6th study site in Michigan. In this case, we combined release and control plot together. So this basically shows you the current ash tree status in our study site. The ash crown class 1 to 2 means the healthy class. Okay, these are all healthy trees. And a different color represents the size of the trees. Okay, at all 6 study sites, you see abundant ash saplings. Okay, this is the number of the ash saplings per 100 square meters based on Transact survey. As you can see, the large trees with DBH more than 20 centimeters in all the study sites are very low. This is because all the large trees were killed by EEB a few years ago. Okay, but there's really a lot of ash saplings as well as some small trees and even some medium-sized trees. The DBH 10 to 20 centimeters. So as of now, I think we are optimistic. At least in Michigan, not all the ash trees are dead. We still have abundant ash saplings and small to medium trees in our study site. And our introduced bowel control agent, in fact, has played an important role, okay, in keeping Amidash-Bower density low and protecting our ash trees to allow the ash trees saplings to regenerate and recover. So I want to quickly summarize what I talked about. So there are 4 parasitoid species were introduced to the U.S. between 2007 and 2015 for biocontrol of the Amedash borer. 2 of the 3 species introduced from China, Obius agrila and Tetrasticus planipanense, have established self-sustaining populations in most release sites and began to play an important role in reducing the Amedash borer densities. You did not hear me talk too much about the other introduced Chinese parasitoid species agrila, because we did not recover any species agrila in our study site. And also studies from other locations also showed that that parasitoid, for some reason, could not establish well, at least in northeastern, northwestern, even in Atlantic states. The 4th species glina from the Russian Far East, was approved for release in 2015. May be complementary to Tetrasticus planipanense to protect large estuaries, because this parasitoid has a much longer ovipositor and can reach the EB larvae in large estuaries. What is really promising is that there are abundant ash saplings survived the initial Amedash borer invasion in Michigan, and now are in fact protected by Tetrasticus planipanense. The egg parasitoids species agrila also established. However, its importance in terms of reducing EB density is still being have to be evaluated, mainly because so far none of the studies have been able to estimate the EB densities, egg densities. So without knowing EB egg density, it's very hard to assess the impact of the egg parasitoid on the EB population dynamics. So far, it seems like Tetrasticus planipanense is a very effective biocontrol agent in attacking, in killing Amedash borer larvae in smaller trees or saplings. Okay. I think that's all I have to say, and I'll be glad to answer your questions. So we have about 12 minutes for questions. Thank you.
Alrighty, we already have one in here. Have ecosystem risks for release for the introduced wasps been considered, and if so, what are they?
Yes. All these parasitoids have been tested for their host specificity. USDA APHIS has a dossier, a document, before issuing permit, outlined their potential risks and benefit of this biocontrol agent. For the 4 agent approved, Tetrasticus planipanense, the one that has established very well so far, only attacks Amedash borer in a lab test as well as in the field surveys. The egg parasitoids, Obius agrila, can in fact attack some of the native blue-breasted beetles' eggs. However, USDA APHIS decide, weigh the risks and benefits. So they think the egg parasitoid, in fact, can provide a great benefit, so outweigh the risk. The risk is some of the native blue-breasted beetles' eggs may be attacked. In a lab study conducted by Dr. Leah Bauer at Forest Service, did show that at least 2 species of the congener agrila beetles' eggs, bronze birch borer, I forget the other one, actually are susceptible to attack by the Obius agrila, by the egg parasitoid. The Chinese larval parasitoid species agrila, and also the Russian species agrila are relatively host specific. They only attack wood-boring larvae that in the same genus Aguilas, only one actually red good spotted oak borer, which is a very serious invasive pest in California. These are the lab study. So there are specificity seem like it's very restricted to attacking the genus Arguellus larvae if there are any risks. The other thing I think if it's considered these wasps do not sting people. Okay, they could not even recognize the naked EEB larvae, so they co-evolved with Amedash borer larvae over millions years. They kind of tuned to the ash volatiles and tuned to the trees that infested with Amedash borer larvae. Yes, the risk assessment was conducted prior to the approval by USDA APHIS.
Okay, we have another question here. Are there climate or phenological indicators for proper timing of this biocontrol?
Yes, there are climate matching before their introduction based on their collection site. There's a software called Climax. We did conduct the climate matching. There seems the one species Grilla, the one that have not established in Midwestern and Northeastern U.S. It turns out that climatically it did not really do well in Northeastern U.S. because it was collected from Tianjin, China. It's much southern than the other species of parasitoids. I think the key in terms of like climatic matching is a synchronization of the parasitoid with Amedash borer susceptible stages, particularly for the egg parasitoid, because Amedash borer eggs will not be only available during a certain period of time, like June, July, and began decreasing in August. That's probably one of the reasons not all the sites released with the egg parasitoid have the successful establishment. So when do you release it? And the other complication is that some of the parasitoid have diapause behavior. And for example, the egg parasitoid. If you release them very early or very late, there's no food to eat, and they basically die. Or you release them in the middle of the season, they find the food, but their progeny come out, and they did not lay the diaposed eggs. They keep looking for food, and the food are gone. So the synchronization with the host stage, it's very, very important in terms of a successful establishment of this agent.
Okay. Are there any predators here in the U.S. of these biocontrol agents?
You mean predators of this biocontrol agent? I think there are actually woodpeckers. Woodpeckers removal of feed on Amedash borer larvae. Actually, we observed too. They also remove the parasitized larvae too. That's the only predator we observed for this parasitoid, for this biocontrol agent.
Okay. What studies have been done to quantify what happens to the EAB population in an area once the major population wave has moved through?
Okay. So far, the only long-term study conducted in Michigan, as I presented in the figure. That study, as of this year, will be 9 years. So the goal is to collect the data at least for 10 years. That's the only one long-term study I know of. I guess because of the funding, and also it's difficult to really maintain a project for over even a few years. The data so far we collect in Michigan, as I presented here, actually that's the only study showing the EEB population crashed. So the key, I think, right now we are really dealing with the aftermath forest. It means following the crash of EEB population, so whether we can really maintain EEB population law for next decade or even longer through reconnecting EAB with its co-evolved natural enemies and to allow our ash trees to recover or regenerate. So in Michigan, I don't know if you know some of the earlier predictions by Dan Herms and group at Ohio State University, and they predict basically all the ash trees, probably 99% of ash trees going to be killed by Amedachi borer. In fact, in Michigan, it's not that case at all. As shown in the last slide, we still have medium, at least medium-sized larger trees. Some, but most small to saplings and small to medium-sized trees, very, very abundant. So our hope that this biocontrol agent will continue to interact with the low EEB populations in Michigan or elsewhere, and so that the saplings can have enough time to grow, recover to larger ash trees.
What range can the parasitoids survive in?
Can you say it again? Sorry. What
range? Yes. What range can the parasitoids survive in?
You mean the geographical range or
I'm guessing that's, yes, geographical range.
Yeah. If you look at what happened in Northeast Asia, each geographical range seems like it has their own complex of this parasitoid. Like in Russian Far East, the dominant parasitoid species is Spithic galena, the large larval parasitoids. But if you move a little bit south to China, North China, the dominant parasitoid is Spithius, it's Tetrascus planpanisa. If you keep moving south, the dominant species parasitoid is Spithic galena. So it really depends, it changes. I guess for the species we introduced, in general, they are northern species. Except the Spithic galena, they are probably can survive, up to Missouri, but I really don't know if we go beyond, like Louisiana, Georgia, because those are northern species. That's one of the reasons I think we should really go back to the native range of Amedachi borer to begin look for some parasitoid species that are more adapted to the southern climatic ranges. So as of now, I think they probably should have no problem surviving Northeast, Midwest, as south as to maybe Missouri, because what's really interesting is even Amedachi borer in its native range, they do not spread as south as like here in the U.S. in Georgia. If you look at the latitude, they probably spread up to like Missouri. That's it, and it's very hard to get any Amedachi borer in southern part of China. So there's still some research that have to be done to figure this out. Okay.
Okay, you're gonna have to bear with me on the scientific name here. Could Tretostachys may be effective in the initial attack of EAB on smaller or thinner barked branches of the upper canopy of large trees?
Yes, they can. However, what's the killing tree in the large tree is the one actually in the main trunk. They are able to attack Amedachi borer larvae on the branches, small branches. But that would not protect the larger trees, because what happened at least we see in Michigan, a lot of EEB larvae is in the main trunk.
Okay. Does your study imply that ash trees will become more susceptible to EAB as they get larger?
Not really. We did not really look at. We didn't really look at the susceptibility. We only look at the protection of the ash trees by Tetrascus planpanisa will kind of decrease as they grow larger beyond the DBH, like 12 centimeter or large trees.
Okay. How big do ash trees need to get before they become reproductive? So if the smaller trees survive, do they at least get big enough to reproduce before they get killed by EAB, allowing the ash species to not go extinct?
Um, I'm not a botanist, but I think I did observe some ash trees with like a DBH 10 to 12 centimeters, uh, some new sprout in Michigan. In fact, they do produce seeds within that range that can be protected by Tetrasticus plantapanisa. But now we are really working hard to try to establish the Russian parasitoid. So hopefully, we'll get something to protect the larger ash trees too. Are we done with the time?
Sorry, I was responding to a question. Okay, is parasitoid rearing keeping up with current demand?
I think I did not talk about the parasitoid rearing. It is a very labor-intensive process, but they seems like at least last year I know they kind of limited, especially with the Russian parasitoid. Last year they were only able to produce like 10 or 20,000 wasps. The rearing are done by USDA Aphis lab in Brighton, Michigan. So overall, the rearing process is very labor intensive because we cannot artificially rear Armitage borer, the host. So all the Armitage borer larvae, adults collected from the field, and then the eggs and the larvae reared in a lab. So we do not have artificial diet to rear Armitage borer larvae. Based on what I know last year, yes, their demand their rearing is limited.
What do you recommend for protection of new saplings post-EAB infestation?
I would recommend to release Tetrasticus plantapanisa if you can and get this parasitoid established in your forest. And then at the same time, you can, when the ash sapling is getting larger, you can try to establish a larger parasitoid. But as we showed in our Michigan study, Tetrasticus plantapanisa is in fact very effective in terms of protecting small sapling trees. But again, there's a climatic issue here. This parasitoid originated from northeastern part of China, so they are really kind of I consider them a northern species. Whether they would do well in a southern U.S. state, I'm not sure. I have not seen any studies on this.
All right, it looks like at this time we don't have any more questions. We've gone over an hour, so it's up to you whether you want to keep going or
I'm done. I need to take a break.
Alrighty.
Okay. Bye.